Assalamu alaikum everyone. My name is Shahd Alheijani. I'm a fourth year medical student, and today I'll be giving you a session on iron metabolism. And inshallah, it's going to be a short session and interactive, and you'll have a few questions in the end, and we can get started. The contents of this presentation, we'll start by a general overview about iron metabolism, and then we'll get to the iron cycle itself, and then how we regulate the iron cycle, and then certain diseases that can arise due to problems in iron metabolism, and finally we have a set of questions. تمام, starting with the overview about iron in the body in general. تمام, هلا starting with what is iron? Iron is a mineral and it's important for certain reactions in the body. So, for example, iron is needed in heme, which is found in hemoglobin in the blood for oxygen transport in the body. It's also found in cytochrome, which is an enzyme which is needed for energy formation in the body. So, for example, cytochromes are found in electron transport chain and in certain reactions which will help form energy. So now, where do we find iron in the body? So as you can see, it's found in different places. So mainly, it's found in hemoglobin. 68% of the iron is found in hemoglobin, which is in the blood. We also have 18% in ferritin and hepcidin. Now, we'll get to details about what ferritin is later on in the presentation, but for now, I want you to remember that ferritin and hepcidin are the iron storage, are for iron storage. Hemoglobin is in the blood. Then we have ferritin and hepcidin, 18% as iron storage, and then we have myoglobin. Myoglobin is found in the muscles, so we have iron either in the blood or stored in ferritin, or as myoglobin in the muscles, and we have 3% as enzymes. So enzymes will include peroxidases, cytochrome, riboflavin enzymes, and so on. Now we also have less than 10%. Can you see my cursor? Let me use the laser pointer. 1 second. Okay, this is better. So we have less than 1% found as transferrin in the plasma. Now, we said ferritin is the iron storage form. Now, transferrin is for transport. So for now, remember that transferrin is for iron transport, and it's less than 1% and found in the plasma. We have ferritin as 18% as iron storage, and then we have functional iron, which is the hemoglobin for oxygen transport, myoglobin in the muscles, and enzymes. Tamam? Now, points to remember. We said iron is a mineral that is needed for energy synthesis in cytochromes and in oxygen transport in the blood. And it's also the star of Fenton reaction. Now, Fenton reaction is, as you can see, this is the equation. So in Fenton reaction, we have iron, which is Fe2 plus, reacting with hydrogen peroxide. It will give us iron Fe3 plus and a hydroxyl radical. Now, hydroxyl radical is a highly destructive reactive oxygen species. Now, the reactive oxygen species will attack the proteins, they will attack the lipids, and so on in the cell, and they will cause cell destruction. Now, sometimes we do need the reactive oxygen species in cases of diseases, but mostly they're destructive for the body, and we don't want that to happen. So this is Fenton reaction. Also, iron is mainly recycled in the body. So what does that mean? It means that iron is not excreted that much in the body. So we try to recycle the iron and reuse it. So only less or a small amount is needed from the diet. But we're recycling the same iron we take in the body. And the minor excretion or minor loss of iron is in exfoliated skin or in exfoliated hair and so on. And iron is either found as ferric or ferrous form. Now, here you can see, so we have Fe2 plus and Fe3 plus. Now, to remember the difference between ferric and ferrous, you can remember S looks like flipped number 2. So ferrous is the Fe2 plus and ferric is the Fe3 plus. Now, we need iron to be in the ferrous form to be absorbed in the body. So ferrous is Fe2 plus. So we need it to be Fe2 plus to be absorbed through the DMT1 channel. We'll get into details about the channels later on, but for now, I just want you to remember that it should be in the ferrous form to be absorbed, but most of the dietary iron is in the ferric form, in the Fe3 plus form. Now, okay. Now about the dietary iron. We can either get iron from the plant sources or from animal sources. Now, animal sources such as red meat have heme iron. Heme iron is more readily absorbed compared to the plant sources. Why is that? Now, heme iron, when we talk about the iron cycle, you're going to see that heme iron needs its own channel. It's not going through the same process as the ferric and the ferrous iron. So heme iron has its own channel, and it's not affected by other things we take in the food. So, for example, we have the oxalate and the phytate I'm going to talk about in a bit. So those will affect the iron absorption. But heme iron is more readily absorbed compared to the plant sources. Plant sources such as the legumes, such as the dark leafy vegetables, are mostly ferric, and we said we need iron to be in ferrous state to be absorbed in the body. Now, what increases and what decreases iron absorption? Now, to increase iron absorption, we need not need, but it is facilitated through an acidic environment. So, in acidic environments such as through gastric acid, through acidic food, and vitamin C, this will help increase the ferrous iron. So, the ferrous iron is the Fe2 plus, which is the one that is absorbed. So this acidic state will increase the ferrous iron, but only 1 to 2 milligrams is absorbed because we said most of the iron is recycled in the body. So most of it is recycled from iron we already have stored in the cell, and only 1 to 2 milligrams is absorbed. Now, this amount that is absorbed may be enough for males, but sometimes females will need more iron. So they have less iron in the body and they need more from the diet. Can anyone tell me why do females need more iron? We have one answer. Okay. Yes. Perfect. Yes. So more loss through menstruation. So because more iron is lost or more blood is lost in menstruation, females will have more iron need. So this 1 to 2 milligrams may be sufficient for males, but iron need is more in females. And someone asked me to repeat a point, but I'm not sure which point it is. Can you please let me know? I think Amina, which one is absorbed more? Yes. So we said heme is more readily absorbed in the body, which is from the animal sources. Um, And okay, so we're gonna get into details in the iron cycle itself. But for now, I just wanted to remember that plant sources have mostly ferric compared to ferrous, so Fe3 plus, and we need to convert it into Fe2 plus, and it's affected by certain things we take in the diet. Meanwhile, in animal sources, we have heme iron. It's a different form of iron that's more readily absorbed. تمام? So, تمام. Okay, we have one more question, but I'm gonna answer it now. So we said iron absorption is increased through vitamin C and acidic environments such as gastric acid and acidic foods. Now, what decreases iron absorption is things that bind to iron and they chelate the iron. So we bind the iron and we prevent the absorption. So this prevention is done through oxalates, phytate, which is found in tea and coffee, phosphate, and calcium. So those substances will bind to the iron and they will decrease the iron absorption, so it's excreted from the body. So how can we apply this? So for example, whenever someone is iron deficient and they have to take iron pills, it's usually recommended that they take this iron with a glass of orange juice, for example, because orange juice has vitamin C. Vitamin C will help absorb the iron because it facilitates the ferrous iron. So now we have more iron absorption. Meanwhile, for example, drinking tea, this will decrease the iron absorption because the phytate will bind to the iron and increase its excretion in the body. Now um, we have more factors. So, for example, we said heme iron is more absorbed in the body because it's from animal sources and it's not affected. But the inorganic iron is affected by several things we take in the diet. We said ferrous form is more readily absorbed, and the acidic environment through vitamin C and acid and solubilizing agents, so amino acid sugars, but phytates and phosphates in tea, those will precipitate, they will bind to the iron and facilitate its excretion instead of absorption. We also have something called hepcidin. Hepcidin, we will cover it again in the iron cycle. Everything is going to come and fall into place in a bit. But for now, just remember that hepcidin will block further iron absorption. So when we have hepcidin, we'll stop absorbing iron. And we talk about it in details in a bit. Erythropoiesis. Now, in erythropoiesis we said, or as you know, it's the process of forming red blood cells. So to make red blood cells, we need to make hemoglobin. Hemoglobin will have heme. Heme needs iron. So when we want to make red blood cells in erythropoiesis, we need iron. So when there's decreased erythropoiesis, we're not forming as much red blood cells. So now we don't need iron as much. So we reduce iron absorption. Meanwhile, when the body or the bone marrow is trying to form red blood cells, we need more iron, and especially when there's ineffective erythropoiesis. So we're making red blood cells, but they're not capable of binding oxygen and delivering oxygen to the body. So the bone marrow is trying harder to make red blood cells. We're producing more, and we have extra iron need in the body. So we need more iron absorption. Same in pregnancy, because in pregnancy, now the mother is not only taking iron for her body, but for two bodies, for herself and for the baby. So now we have increased iron absorption. And finally, hereditary hemochromatosis, which is basically a condition in which the body is taking too much iron. So we have iron overload in the body. And we're going to cover it in the end again. So this is just a general overview. And now we can move on to the blood tests. So we can carry different tests in order to measure the amount of iron in the body. Now, before going into the blood tests, does anyone have any question about the previous part? Mhm. Cytochrome. Okay, so you don't have to know cytochrome details, but for now, just remember that cytochrome is an enzyme that's needed for redox reactions. So it's responsible for reduction and oxidation of certain compounds. And cytochrome needs iron. So iron is found in certain enzymes, including cytochrome, peroxidase, and so on. But for this lecture, you don't have to know the function of cytochrome. It's found in different reactions. For example, in the electron transport chain, you'll have different cytochromes that will help in the redox reactions to, in the end, to form energy for the cell. So, okay. So no more questions, so we can move on to the blood tests. Now, for blood tests, we have four main things we measure. So we have ferritin, we have the serum iron, we have the total iron binding capacity, or the transferrin, and finally transferrin saturation. So, starting with ferritin. So we said ferritin is the intracellular iron storage. Remember when we mentioned where we find iron, we said here ferritin is the iron storage protein, iron storage form. So here, serum ferritin will correlate to the total body iron stores. So iron that is found inside the cell, stored in the cell. And it's the most sensitive test for iron deficiency anemia. So I want you to remember that in iron deficiency anemia, the first value that will go down is the ferritin. Because ferritin is the iron source. And now when we have iron deficiency anemia, we're trying to release the iron that's trapped inside the body, inside the cells, in order to utilize it. So the first thing that will go down in iron deficiency anemia is ferritin. تمام. Now we have serum iron. Serum iron is just the total circulating free iron in the blood. So again, in iron deficiency, you'd have less serum iron, for example. Total iron binding capacity, or transferrin. We said transferrin is the thing that is responsible for the transport of iron. So you can see those blue circles. This is transferrin. So it binds to the iron and it transports it in the blood. And an indirect way to measure transferrin is the total iron binding capacity, because we said this will the transferrin will bind to the iron and will move it in the body or in the blood. And finally, transferrin saturation. So transferrin saturation is the amount of iron that is bound to the transferrin, and this is calculated through serum iron dividing serum iron over the total iron binding capacity. You don't need to know the details about this or the normal values, because they usually provide the normal values. But I have everything written in the notes section under the PowerPoint. I'm going to send the PowerPoint as soon as I'm done. And if you want to know, you can find them in the notes section. So in iron deficiency anemia, we said the serum iron will be low and the ferritin will be low because it's iron stored in the cell and we need to release it. Now, what about transferrin? What would happen to transferrin in iron deficiency anemia? Can you type in the chat? So, more answers? Perfect. Exactly. Clear. So the transferrin levels would rise in iron deficiency anemia. So we said ferritin and serum iron would decrease, but transferrin would rise because now we're trying to take to absorb more iron and take it into the cells. So we need more transferrin to transport this iron into the cells. So transferrin level will rise, serum iron will decrease, and ferritin will decrease. تمام. Now we can move on to the iron cycle itself. Any questions about the first part? Okay. تمام. Thank you. Right. Now to the iron cycle. So this is a general overview about the iron cycle. As you can see on the top, we have the mucosal cell, we have the enterocytes, which will absorb the iron from the body, from the diet. So, for example, in the intestines, and then we have the blood, and the iron will go from the enterocytes to the blood, and finally to the target organs, either to the liver or to the blood cells, to the macrophages. So we'll take them step by step. Let's start with the absorption of iron from the diet in the enterocytes. So, number one, enterocytes. Now, for enterocytes, I want you to remember that there's an apical membrane and there's a basolateral membrane. So the apical membrane is the one that faces the lumen. So everything that we absorb in the body passes here, and it will pass through the apical membrane, going down into the basolateral membrane to the blood. So now let's start with the apical membrane. We can either have heme iron, as we said, and the dietary and the ferric iron. So heme iron, we said, mainly comes from animal sources. So it's more readily absorbed. And as you can see, so here we have the structure of heme, and this is the iron bound to heme. So through heme oxygenase, we'll take the heme iron, and then heme iron can pass through the heme transporter into the enterocyte. So it doesn't need all the other stuff for the dietary ferric iron. It only needs this heme transporter. So now for the dietary iron, which is ferric. We said ferric is the 3 plus, ferrous is the 2 plus. And in order to absorb iron, we need Fe2 plus. We need ferrous iron. So mostly we have it as Fe3 plus. Now, first step we need to do in order to absorb iron is to change it into the absorbable one, which is the Fe2 plus. So this is a reduction reaction. So we need Fe3 plus to become Fe2 plus. And what is responsible for reduction? The duodenal cytochrome B. So D-cytochrome B or perireductase. This is the enzyme that is responsible for the reduction of Fe3 plus to Fe2 plus. So now this is the absorbable form. Now we converted it into a form that can go into the cell. Now we need to transport it into the cell through a transport or a channel that is called DMT1, which is the divalent metallic transporter 1. So now we converted it into Fe2 plus. Fe2 plus can now go into the cell through the DMT1 channel. This is in the apical membrane. We took it inside, and now we have Fe2 plus inside, either from the heme or from the diet, from the iron. Now we have it as Fe2 plus. Now we can store it inside the cell. So storage form of iron, we said, is ferritin. Now ferritin has ferroxidase activity, so it can either convert Fe2 plus to Fe3 plus and vice versa. But for now, I just want you to remember that ferritin will store the iron inside the cell. تمام? Now, what is the effect of hypoxia? Hypoxia, in other words, we have less oxygen. So you're taking in less oxygen, and there's less oxygen reaching the cells. So now you need to compensate. You need to increase the blood going into the areas, increase everything, so that more oxygen can reach the cell. And in order to do that, you need more iron. So you can have so to increase the red blood cells, to increase everything, in order to compensate for this hypoxia. In order to do that, you need to increase the amount of transporters and channels and enzymes that will help you absorb this iron. So this means that in hypoxia, we will upregulate the duodenal cytochrome C and upregulate the DMT1 channel. So hypoxia will upregulate these two in order to increase iron absorption. تمام? So this is for the apical membrane. Now we can move on to the basolateral membrane. Now we took everything. We took the iron. It's now stored in the enterocytes inside the cell. Now we need to utilize it. We just absorbed it and it's still in the intestine. We needed to take it to the target organ. Now, we will take the iron stored in the ferritin and release it into the body. Now, to release it to the blood, we need something called ferroportin. So it's a port. The port will help release outside. So we'll take this iron, which is still in the Fe2 plus form. It will leave through the ferroportin into the blood. Now we have a problem because we said the transport form of iron is transferrin, صح? So now ferritin stored the iron. Now the iron is released through ferroportin, but it's still as Fe2 plus. Transferrin can only bind iron if it's in the ferric form, Fe3 plus. So we said to be absorbed, we needed Fe2 plus, which is the ferrous form, but to bind to transferrin, we need it as ferric. Now we need something to oxidize this ferrous to ferric form, which is the hepcidin. Hepcidin is the enzyme, again, on the basolateral membrane that will help oxidize the Fe2 plus to Fe3 plus. So now the ferric form can bind to transferrin. So we said we have the ferroportin and the hepcidin. Now we have the ferric iron. Now it can bind to apotransferrin. So apotransferrin is before binding. Now when it can bind to the ferric iron. So it can take two. Now when the two bind to the apotransferrin, it's now called diferric transferrin or holotransferrin. So here you can see we have the holotransferrin. So we said first we have the duodenal cytochrome B enzyme to get the ferrous iron through the DMT channel. It goes inside, but for the heme iron, it only needs the heme transporter. Iron is stored as ferritin. Now it can leave through ferroportin, but it's still in the ferrous form. It's converted to the ferric form by oxidation through hepcidin, and now it can bind, two of them can bind to one apotransferrin to form holotransferrin. So far clear? Yes, so ferritin will store the iron. Now, for now, it's stored as Fe2 plus, تمام? And it's still in the intestines. Now, in the intestine, in the interior side, we said it's absorbed as Fe2 plus and still stored that way. But in other cells, you're going to see it will be in the Fe3 plus form. But for now, I just wanted to remember that ferritin stores the iron. You don't need to go into details. Just remember ferritin, iron storage. Transferrin, iron transport. تمام? Okay. Now, Hepcidin. Okay, we're absorbing iron. We said we have iron going through the intestine to the basolateral membrane to the blood. Now, if this process keeps going on, it keeps continuing, we'll have too much iron in the blood. So we need a mechanism to stop the iron absorption in case we have too much iron. So this is done through hepcidin. Now you can see we have high iron. We have too much iron binding to the transferrin. Now transferrin will bind to the transferrin receptor in the hepatocytes in the liver. Now we have transferrin receptor 1, transferrin receptor 2, it's binding. And the hepatocytes can now sense that we have too much iron because we have a high amount of iron in the blood. So it will release something called hepcidin. Hepcidin will help stop the iron absorption by blocking ferroportin. So now you can, if you want to remember what it will block or its action, you can think of hepcidin as security guard. Security guard where? In an airport. So this security guard will go to the port, to the airport, ferroportin, and will block it. So hepcidin will block ferroportin or close ferroportin. So now this iron can go from the enterocytes into the blood. So hepcidin will be upregulated when we have high iron stores. And this is what we mentioned in this table. So we said increased serum hepcidin will help us reduce iron absorption. But if we have less hepcidin, we can normally carry on absorption because we have the ferroportin channels open. So this is the function of hepcidin. To block ferroportin, block iron absorption, we have too much iron now. Now, we need something to regulate hepcidin itself. We have hepcidin blocking the ferroportin channel to stop iron absorption. But what if now we need iron? And we have a situation in which we need iron and we have hepcidin. So we need something to regulate this hepcidin. And this thing is called erythroferrone. Erythroferrone is formed from erythroblasts. Now, erythroblasts are the precursors of red blood cells in the bone marrow. So you can see in this diagram, here are the erythroblasts in the bone marrow, and they would release erythroferrone. erythroferrone will go to the liver and decrease hepcidin. Why? So we have erythroblasts in the bone marrow, and we want to form red blood cells. So to form red blood cells, we need to complete the red blood cell. We need heme, and heme will need iron to finish the structure, so it can be able to bind oxygen, to transport oxygen to the body, and so on. And to get this iron, we need to absorb it from the intestine. And if we have hepcidin blocking the absorption, we can't form red blood cells. So the erythroblast will release erythroferrone. Erythroferrone will decrease hepcidin. So now we can absorb iron from the duodenum and we can form the red blood cells. تمام? So erythroferrone is released from erythroblasts. It will decrease hepcidin production, so more iron is absorbed, and it's regulated by erythropoietin. Erythropoietin is released from the kidneys. Now I want you to remember that erythropoietin is released from the kidneys. So erythropoietin will act on the bone marrow. It will stimulate the erythropoiesis basically. And now the erythroblast will release the erythroferrone. So erythropoietin from the kidneys stimulate the erythroblast. They will release erythroferrone to the liver and now will decrease hepcidin production so we can absorb the iron. Iron is absorbed and now we can form red blood cells. So when do we have more hepcidin and when do we have less hepcidin? We have more hepcidin in conditions where we need less iron absorption. So we need to stop iron absorption. So for example, if we have high transferrin saturation, when we have high transferrin saturation, that means that we're absorbing so much iron. So now we need to stop it. That will lead us to increasing hepcidin production from the liver. Also, other cases such as inflammation. So in cases of inflammation, we're going to talk about inflammation at the end of the PowerPoint when we talk about the pathologies. But inflammation will cause an increase in hepcidin. So now we block iron absorption. So when we have a high transferrin saturation and inflammation, we have high hepcidin. We have less hepcidin in case of anemia and hypoxia. So for example, I want someone to tell me what happens in anemia. What is anemia? Mhm. Okay. So in anemia, we have less, yes, total hemoglobin. So yes, exactly. تمام. So in anemia, we have less red blood cells or less hemoglobin in the body. So now we need to compensate for that. We need to increase the red blood cell production. We need to increase erythropoiesis. To increase erythropoiesis, we need iron because iron is needed in heme. Iron is needed to form hemoglobin. So in cases of anemia, we do not want hepcidin to get in the way of iron absorption. So we decrease hepcidin, and same in hypoxia. So in hypoxia and anemia, we decrease hepcidin production because we need to increase the amount of iron absorbed from the body. Is that clear? Yes. So, okay. Now we have a question. What happens if hepcidin is deficient? What if we do not have enough hepcidin? What would happen? Yes, we'll have iron overload. So if you do not have enough hepcidin in the body, we'll keep on taking the iron, we'll keep on absorbing the iron, and this will give us iron overload. And we see iron overload in different conditions. So for example, we see it in hereditary hemochromatosis. Again, we're going to talk about hereditary hemochromatosis at the end, but for now, I want you to remember that hemochromatosis, you have so much iron in the red blood cells or in the body. When you have increased iron in the body, this means that you do not have enough hepcidin. So, for example, in case of hereditary hemochromatosis, there is a gene mutation. We'll talk about this in the end, but this mutation will cause increased absorption. So if you have decreased hepcidin or you have iron overload, this will cause too much iron to be in the body. تمام? But in cases of inflammation or high transferrin saturation, you'll have more hepcidin release because we're trying to block the iron absorption. So this is for hepcidin regulation. Now we finished the absorption through the enterocytes. Now the iron is found in the transferrin. Transferrin is moving in the blood. Now it needs to go to the target tissue. Target tissue now will have the transferrin receptors. We have the transferrin in the blood. Now the transferrin needs to bind to its receptor in order to be absorbed. So here you can see this is the cell membrane for the tissue. And you can see you have transferrin receptor 1, transferrin receptor 2. This is the transferrin, the whole of transferrin, or the dipheric transferrin, with the iron attached to it. And what form of iron did we say we have in transferrin? Is it the ferric or the ferrous? So, yes, in the Fe3 plus. So it's ferric here. Someone asked what happens if there is too much iron. Okay, again, we're going to go into details about this at the end of the presentation and the pathologies. But for now, let's just think about the physiologic state, the normal state. And we'll talk about the pathologies in the end. Tamam, yes. So transferrin will have it in ferric state. Now, the transferrin will bind to the transferrin receptors. And now here we have something called a clathrin-coated bit. So clathrin will help us in the formation of an endosome. So we have transferrin binding to the receptor, and now an endosome will be formed by the clathrin-coated bits. Now here we have the transferrin receptor, we have the transferrin, and we have the ferric iron. Now we need to release this iron from the transferrin in order to take it into the body. How do we do that? We need an acidic state inside the endosome. Now, to get an acidic state, we have the hydrogen, we have the proton pumps. So hydrogen will go into the endosome. This will acidify the endosome. So now, as you can see here, the pH is low. pH is 5.5. Now, this will help us detach the iron from the transferrin. So now the iron is released from the receptor and from transferrin. Now it can be pumped outside through the DMT1 channel. So we mentioned DMT1 previously here. So this will help us take it into the cell. Same case here, DMT1 is taking it into the cell. DMT1 take iron into the cell, and now this endosome can be recycled. So it will go and attach again to the cell membrane, releasing the apotransferrin, because we said apotransferrin is not bound to iron. But holotransferrin has iron. So now we have apotransferrin, and the transferrin receptors are recycled back to the surface. Now iron, we have it inside the cell. Is it clear? Any questions? Are we in enterocytes? No, this is not in the enterocytes. So this is the enterocytes. So we said the enterocytes will help with absorption. Now it's absorbed, it will go into the blood, bind to the plasma transferrin. Now this transferrin will take it to the organs. So we'll take this iron into certain organs. So the target organ could either be the liver. So we have the hepatocytes. It could be in the red blood cells. It could be in the macrophages and so on. But for now, this is the enterocyte. We absorb the iron, it went to the transferrin. A transferrin will take it to the target organ. تمام? Okay. Released iron is still in the ferric state. It's not converted yet. We said we needed the Fe2 plus to be absorbed in the enterocyte. But for now, خلاص, we have this as ferric, Fe3 plus. تمام. No more questions, we can move. Now, to store the iron, we said iron transport is by transferrin, and iron storage is ferritin. So here we have something that's like a cage or a jail in the cell, and it's called apoferritin. So when iron comes inside, now we can store it inside the cell in ferritin. One ferritin can bind up to 4,000 ferric or the iron molecules, basically. So as you can see, this is a macrophage and this is a hepatocyte. So those may be the target organs or the target cells. So those cells will have the iron stored inside. It's stored as ferritin, stored in ferritin, in hepatocytes and macrophages. Now, when it's stored, we can't utilize it. It's locked inside the cell. But what if we need this iron? Now we will degrade the ferritin. We'll break down the ferritin by lysosomes. So the lysosomes will break down the ferritin in order to release the iron. Now, what would happen to the ferritin itself? Ferritin is partially degraded. Now, when it's partially degraded, we call it hemosiderin. And remember, again, when we talked about the iron in the blood, iron in the body, we said ferritin and hemosiderin are the iron storage proteins. So hemosiderin iron storage molecules. So hemosiderin is partially degraded ferritin. So when we need to utilize this iron, we break down the ferritin. Partially degraded ferritin is now called hemosiderin. And as you can see here, you can see this kind of brown color. This is the hemosiderin. So in some diseases, in heart failure, for example, or in any diseases in the lungs, you can see macrophages engulfing the red blood cells And now when the red blood cells are broken, now we're taking the iron out. Now we're breaking down the ferritin to take the iron out, and the broken ferritin is now called hemosiderin, and it will give you this color. Obviously, you don't need to remember this picture. This is just to visualize it, to remember it. What is epiferritin? Epiferritin is just the free ferritin. So, ferritin in the cell, it will become ferritin. Apoferritin will become ferritin, and it will store the iron inside. That's it. So we said the free transferrin is apotransferrin, the free ferritin is apoferritin. That's it. It's not a different molecule. So now that's it for cellular storage. So we talked about the iron cycle itself, from the enterocytes, apical membrane to the basolateral membrane, and then hepcidin blocking the ferroportin, and then how we regulate hepcidin through erythropoietin, and how we absorb this iron into the cell through transferrin and the clathrin-coated bits, and then we store it as ferritin inside the cells, either hepatocytes or macrophages. Can you please clarify this point? Which one? Which point do you want to repeat? Okay. Now we're talking about iron stored in ferritin. It's not doing anything. It's just stored in the blood. In the cells, I mean. So we said we need it as ferrous to be absorbed, and then it goes inside, and then ferric. But now we're not utilizing this iron. We're not doing anything. It's just stored inside the ferritin. So for now, we're not using it. It's not doing anything. It's just trapped or just packed inside ferritin. It's not doing anything. So only when we need. No, they're the same thing. They're the same thing. It's not stored as ferritin. So someone's asking if ferritin gets stored as ferritin. Now, okay. Apoferritin will just bind the iron and form ferritin. Ferritin is a storage molecule. It's not a different thing, and they're not separate. Just like we said, apotransferrin will bind to the transferrin molecule. Transferrin will bind to the iron molecule and form a holotransferrin. So it's not a different entity. تمام. طيب. Now for the regulation of the iron cycle. Now, how do we take care of the iron cycle? Now we said we have hepcidin regulating the amount of iron absorbed from the enterocytes, and then we have the erythropoietin regulating hepcidin. But how do we regulate the cycle as a whole? Now we regulate it through iron response proteins. Now, iron response proteins can either be free or bound to iron. So when an iron replete cell means that we have enough iron in the cells. So iron will bind to the iron response proteins, and it's not going to have any action. But now in iron deficient cells, this will have an action. It will do something. So what will it do? Iron response proteins will bind to the iron response elements on the mRNA. So how do we regulate the iron cycle or the iron metabolism? Through post-transcriptional regulation. So after transcribing, now we have the mRNA. Now we can regulate this mRNA through iron response protein binding to the iron response elements. Now it can bind in different locations. It can either bind to the 3' untranslated region or to the 5' untranslated region. So it will have different action when it binds to those regions. It can either bind to the 3', and this will lead to increased synthesis of transferrin receptor 1 and the DMT channel. Why? Now, we have less iron in the body, so we need to absorb this iron, and we need more iron. And in order to do that, we need more of the DMT channel, because we said the DMT channel is the one that will take this ferrous iron and take it inside the cell. So we need more of this because we need more iron to go into the enterocyte. And we also need more transferrin receptor. So we need more transferrin receptor because this absorbed iron, we need it to bind to transferrin to go to the target tissue. So we need more of this. So when the IRP binds to the iron response element in the 3' untranslated region, it will cause an increase in protein synthesis of transferrin receptor and the DMT1 channel. Meanwhile, what would happen if it binds to the 5' region? So if it binds to the 5' region, it will decrease the translation of ferritin and ALA synthase. Now, ferritin, we said, is the iron storage protein, iron storage molecule. Now, if we do not have enough iron in the body, we cannot store iron. We need to mobilize this iron. We need it to be utilized either in enzymes, in forming red blood cells, and so on. So we can't afford storing this iron, so we need less ferritin. So it would reduce the translation of ferritin and also the ALA synthase. Now, this is an enzyme that is needed in a different reaction to form heme and hemoglobin. Now, if we do not have enough iron, we will downregulate the ALA synthase because we do not have enough iron. But we'll also downregulate ferritin because we do not want to store the iron. We need more iron to go into the blood. And how do we make sure we have enough iron going to the target cells? By increasing iron absorption, so you have more DMT, increasing transferrin, so you have more iron going to the target cells. So if you want to remember, okay, it's okay, all good. So remember that the 3' region would result in increased synthesis of certain proteins. 5' region will decrease. But again, we have different proteins in both cases. So in the 3', we have increase of TFR1 and the DMT. In 5', we have decreased, but for ferritin and the ALA synthase enzyme. And finally, we have the pathologies. So we talked about everything normal in the body. And then we're going to move on to the abnormal things. So before that, any questions? Okay. Okay. So yes, binding the iron response protein binding to the 3' region will increase the transferrin and DMT protein synthesis, but binding the same thing, binding to the same to the mRNA at the 5' region will decrease the formation of ferritin and ALA synthase. So we have the iron response protein. It can either, if you have enough iron in the body, it will just bind to iron. It won't have any effect on the mRNA. تمام? But if we do not have enough iron, so it's not bound to iron. Now it will go and bind to the mRNA. It will cause a post-transcriptional modification. So now it can bind to mRNA. It can either bind to the 3 region or the 5 region. The 3 will increase transferrin and DMT. The 5 will decrease ferritin and ALA synthase. ALA synthase is another enzyme needed in a different reaction to form porphyrin, protoporphyrin, and red blood cell synthesis. You do not need any details about that. You can learn it later on in heme. But for now, just remember that the binding to the 5' region will decrease ferritin and ALA synthase. So, any other questions? Okay. Okay, so this is the last section before going on to the questions. And it's just a short section, inshallah. So we can either have iron overload or anemia of chronic disease. Those are the two cases we're going to talk about for now. So, in iron overload, we mentioned iron overload once in hereditary hemochromatosis. So this diagram basically explains everything that happens in hereditary hemochromatosis. So we have a gene mutation. You do not need to remember any details about the pathogenesis itself and the gene mutation and so on. I just wanted to remember that now you have too much iron absorbed and you have iron overload in the cell. This will cause, someone asked about what happens when you have too much iron. So here you can see it. So you have arthritis, you have something in the typical presentation is called bronze diabetes. So you'll have too much glucose in the body, and it's called bronze diabetes because the skin will appear bronze in those patients. Again, this is just a fun fact. You don't need to remember details. Just remember that you have too much iron now. And also another case of iron overload is in hemolytic anemias, such as thalassemia and other hemolytic anemias. Now, in both cases, you have too much iron. Now, okay, we took this iron, we utilized it in enzymes, we utilized it in myoglobin, in hemoglobin, we stored it, and still we have too much iron. What would happen to this iron? This iron will go through something called Fenton reaction. Remember we mentioned in the beginning, Fenton reaction is when it happens, it will give us reactive oxygen species, the hydroxyl radicals, and so on. Reactive oxygen species will go and now destroy the proteins, the lipids, the nucleic acids, the cell membrane, and now the cells will start dying. Now the heme iron will also damage the cells in the liver, pancreas, and heart. So when you have too much iron, we utilize everything and still we have excess. It will go through Fenton reaction and it will cause cellular damage. Now we also have something called hemolytic anemias. Now I trust you studied about hemolytic anemias and beta thalassemia, and all those anemias. Can anyone tell me what happens in beta thalassemia? Yeah, a quick review. Briefly. Mm-hmm. Yes. So there's a problem with forming the beta chain. So now we can't form proper hemoglobin. So now yes, exactly. So now the bone marrow is sensing, okay, we're forming red blood cells, but we can't form proper hemoglobin. We can't form the beta chain. So now it's trying to compensate. So this is an example in beta thalassemia, but it's in hemolytic anemias in general. The bone marrow is trying to compensate. It's trying to produce more red blood cells. So now we have more erythroblasts. We said erythroblasts are going to produce a certain substance in order to increase the iron absorption. Can anyone remind me of the substance? Yes. So erythroblasts would release erythroferrone. Yes. So we have more erythroblasts because we're trying to compensate for the hemolytic anemia. We're producing more red blood cells, more erythroblasts. Those would release more erythroferrone. We have less hepcidin. We said erythroferrone will decrease hepcidin, so now we can absorb iron. But in hemolytic anemias, this is happening too much. So now we have iron loading. We have too much iron. And now we have erythroid hyperplasia in the bone marrow. Erythroid hyperplasia is just because we have too much iron, too much red blood cell production, too much erythropoiesis going on. So you can see this bone marrow is hypercellular now. You have too much erythropoiesis going on. But what you need to remember from this section is you can see iron overload in hereditary hemochromatosis, which is a disease in which you have too much iron absorbed. And also in hemolytic anemias, just thalassemia, because the body is trying to compensate, and now we're producing a lot of erythroblasts. And when we have too much iron, this will destroy the cells because iron will undergo Fenton reaction, and now they will produce reactive oxygen species. The other case, which is the last thing we're going to talk about, is anemia of chronic inflammation. Now, what happens in inflammation is the main culprit in inflammation is the macrophages. Macrophages are trying to protect the body. They're trying to fight the inflammation. So they will release certain cytokines, certain chemicals, such as we have TNF alpha, you have interleukins, cytokines. You don't need to remember everything, but for now, just remember interleukin 6. Interleukin 6 is released from the macrophages in case of inflammation and in certain conditions. So, for example, you have HIV, you have tuberculosis, rheumatoid arthritis, and malignancies. You have interleukin 6 released from macrophages. What would this interleukin 6 do? This will increase, it has several functions, but for now we're focusing on the iron aspect. So interleukin 6 will increase hepcidin from hepatocytes. So now we have interleukin 6 from macrophages. It will go to the hepatocytes in the liver, increase hepcidin production. Now, when we have hepcidin, what will happen? Hepcidin will decrease the iron absorbed from the GI, and now the iron is trapped in the macrophages and the hepatocytes. So we have iron in the body. Iron is enough in the body, but it's only trapped. It's trapped in the macrophages, it's trapped in the enterocyte, in the hepatocytes, and we can't absorb more iron from the GI. So now, if we want to make red blood cells, we do not have the capacity to mobilize this iron. Now the iron is only trapped inside the macrophages, hepatocytes, enterocytes. So we said we need the ferroportin to take out the iron from the enterocytes into the body. But now ferroportin is blocked or it's destroyed because of hepcidin. So it's now trapped inside. So even though the patient has enough iron, we're taking in the iron, but we can't absorb it. And the iron is trapped now. So this will lead to less erythropoiesis, causing anemia of chronic disease. So this, when it is going on for a long time, the inflammation is going on for a long time, we have continuous IL-6 released, interleukin 6 released. This will increase the hepcidin. Now the iron can't be absorbed, it can't be utilized, it's trapped. So now we have anemia because we can't form the red blood cells, we can't form the hemoglobin. So this is anemia of chronic disease. You do have the iron, but you can't utilize this iron. You can't absorb it. You can't take it out from the macrophages or the hepatocytes or the enterocytes. So the patient would basically have a chronic disease, for example, SLE or any condition or inflammation, rheumatoid arthritis. So even if they're taking iron, so you can see that the iron store in those patients would be normal, but now they still have anemia. They still do not have enough red blood cells. You take a bone marrow sample, it still does not have enough red blood cells, even though they do have the iron. But because we have IL-6, we have the hepcidin, you can't use this iron, you can't absorb it, you can't take it out. So now you have anemia of chronic inflammation. And that is the end of the lecture. Now we have 3 questions to solve. But if you have any questions before that, please feel free to ask. Or you can keep them to the end. I'm fine with everything. Any questions? I think no questions. Slide 21. Phase 2. Wait. Oh, ماشي. هلا this is iron overload. Iron overload means we have too much iron in the body. Now this can be caused by different mechanisms. For example, a disease called hemochromatosis. In hemochromatosis, the body is absorbing too much iron. Even though we do have enough iron in the body, but we're still absorbing more. And hemolytic anemias, the red blood cells are bursting. The hemolysis means the red blood cells are dying, they're bursting. So in hemolytic anemias, the blood cells are dying, and now the bone marrow is trying to compensate. We're trying to form red blood cells, so it accelerates the erythropoiesis. Now, when we want to do that, we need more iron to be absorbed to form the hemoglobin to form the red blood cells. Now, in order to in order to do that, we're absorbing more iron. So, how does that happen? Because the bone marrow is trying to form the red blood cells. Now we have more erythroblasts. These erythroblasts will increase erythroferrone. Erythroferrone will decrease hepcidin. Now we have iron loading. We're taking more iron. So this would lead to iron overload in the body. And we have erythroid hyperplasia, which means that we have hypercellularity in the bone marrow because we're trying to compensate to for the hemolytic anemia. So in hemochromatosis and hemolytic anemia, we have iron overload. This too much iron will go through Fenton reaction. It will oxidize and give reactive oxygen species, and those will attack the proteins, the lipids, nucleic acid, and so on. Is that clear? Right. And now I have a few questions for you guys. Let's start with question 1. Which of the following regulated proteins is released by RBCs? Easy question. Yes, everyone's correct. It's erythroferrone. طيب, follow-up question. Where is hepcidin released from? From hepatocytes, which are the liver cells. Yes. طيب, one more question. Erythropoietin. We mentioned it in this presentation, but yes. Erythropoietin is from the kidneys. Erythropoietin is released from the kidney, stimulate the bone marrow to produce erythroblast. Erythroblast will produce the erythroferrone. Erythroferrone will decrease hepcidin production from the liver. طيب. Next question. Iron deficiency anemia. Okay. تمام. Any other answer? كله إيه؟ Perfect. Correct answer is A. ماشي. Okay, follow-up question. Which of those, the serum iron, transfer, and ferritin, is going to change first? Ferritin. So we said the most sensitive one is ferritin. It will decrease first in iron deficiency anemia. And we have decreased serum iron because obviously total iron in the body is going to be low in iron deficiency anemia. And we have increased transfer because we want to take this, we want to absorb more. And now this absorbed iron will bind to transferrin in order to be taken to the target cells. So we need more transferrin. Perfect. تمام. Last question. What is the effect of hypoxia on the iron cycle? Alhamdulillah, mashallah. Um, Everyone's correct. Hypoxia will upregulate the cytochrome B, the duodenal cytochrome B. Follow-up question. What does this molecule do? What does it do in the iron cycle? Mhm. Fe3 plus Fe2 plus. So ferric to ferrous. Now, when we change ferric to ferrous, now we can absorb the iron because iron can only be absorbed in the ferrous state as Fe2 plus. Now, why is B wrong? Yes, exactly, yes. So, mhm, yes, we need to down-regulate hepcidin. Yes, exactly. We need to down-regulate hepcidin. We said hypoxia will down-regulate hepcidin. It will up-regulate duodenal cytochrome B and the DMT channel. So when we decrease hepcidin, now we can absorb more iron, and which is, yes, exactly, which is what we need in case of hypoxia. Um, okay, what does hepcidin block? Last question, خلاص. Mm-hmm. Yes. So hypoxia will block ferroportin. خلاص ما شاء الله. I think you guys are ready خلاص. This lecture, I hope I explained everything well. But in case you have any questions, either now you can ask me or later on you have my contact information. You can ask me anytime. Thank you so much for your attention. Anything you need, I'm always here to help, inshallah. Thank you. Well, have a great day, everyone.